Hello, my dear student. Welcome. Today we are going to discuss your biotechnology part. Particularly, we are going to discuss about unit second and unit third in very short because we are more interested in GPAT Niper Drug Inspector Pharmacist. Whatever are the important points for GPAT Niper Drug Inspector Pharmacist, I am going to take only that. I will discuss only that. Okay, so in your syllabus it is given that in case of unit second, various vectors restriction nucleases enzyme DNA ligase recombinant DNA technology and its application, then your production of viral vaccine, bacterial vaccine and hormones. Okay, a brief introduction of polymerase chain reaction. Okay, so first we will see some examples which are important for recombinant DNA technology. Okay, then we will see the polymerase chain reactions in the first video. In second video we are going to discuss about the recombinant DNA technology itself. Okay, so let's start today's session. So first of all, few I will like to discuss few enzyme with you, enzymes and their functions. Okay, so first enzyme, you can see this is a reverse transcriptase. So what is a enzyme? That it is an enzyme that used to synthesize DNA from mRNA. Okay, it is normally in the process of transcription, DNA is converted into mRNA. Okay, so this is a normal transcription. DNA is converted into mRNA. You can remember the central dogma of life, that is a replication of DNA, then transcription, that is transcription means DNA to mRNA, then translation mRNA to protein synthesis. This is the central dogma of life. But in case of reverse transcriptase will make a DNA from mRNA. Okay, so it is going to in reverse direction. That's why it is known as reverse transcriptase. It is reversing the transcription process. So it is reverse transcriptase. Okay, so these enzymes are used to make DNA from RNA, and these enzymes are found in retroviruses. For example, your HIV virus, human immunodeficiency virus, it is also a type of retrovirus. So you can find reverse transcriptase enzyme in that. So please understand it, this reverse transcriptase and its function. Then RNA polymerase, obviously all of you know that RNA polymerase. What is the function of RNA polymerase? It will join RNA nucleotide to make a mRNA during transcription. So when there is a transcription process, RNA polymerase will come into the picture. It will make mRNA from DNA. Okay, then next one is DNA ligase. So DNA ligase, DNA helicase, sorry, DNA helicase. DNA helicase, it will unwind the DNA helix within the molecule. Okay, it means it will separate the two strands of DNA. Okay, DNA helicase will separate two strands of DNA. So next enzyme is DNA ligase. DNA ligase. So DNA, as the name indicates, ligase means to join. So it is the enzyme responsible for joining. Okay, so DNA ligase will join the slices of gene to make the recombinant DNA. Okay, if you have breakdown or slices or pieces of DNA, the enzyme DNA ligase is used to join that pieces of DNA. Then next enzyme is DNA polymerase. So DNA polymerase is it is responsible for the your replication. It will join nucleotides in the DNA replication. Okay, so next enzyme is restriction endonucleases. So there are two types of nucleases enzyme, endonucleases and exonucleases. Exonucleases enzymes are produced outside the cell, and endonucleases enzymes are produced inside the cell. Okay, so we will our important for our point of importance, restriction endonuclease are important. So restriction endonuclease, they are obtained from bacteria, and as the name indicate, restriction, they will cut the DNA at a specific sequence which is known as recognition sequence of that enzyme. So restriction enzyme or restriction endonucleases are used to cut the genes or used to cut the DNA to make a cut into DNA at a specific determined sequence, that is known as recognition sequence. Most of them are the sequence are of palindromic sequence, means ATGCA TTGA. Add the sequence adenine, thymine, thymine, adenine, guanine, like this. Okay, that is known as palindromic sequence. They will cut at the palindromic sequence. Okay, so these are the few example. More example we will see about the enzymes. So here are few different enzymes used in enzyme DNA cloning. Enzymes used in DNA cloning. So alkaline phosphatase. So it is responsible for removal of phosphate group from 5' chain of your DNA or RNA, double helical DNA or RNA. Then DNA ligase, we have already seen what is the function of DNA ligase. It will join the uh, two pieces or two slices of DNA. Then DNA polymerase, we have also seen this DNA polymerase. Then exonucleases, already I have seen told you nucleases enzyme. They have property to break down, to cut. So nucleases will cut down the whatever is your gene or DNA sequence. So exonucleases, it will result in cleavage. So then next one is mung bean nucleases and yes1 nucleases. Okay, so mung bean nucleases, it will digest only single stranded DNA or RNA. Okay, then nucleases yes1, it is also similar to mung bean nucleases, but it will act in reverse direction. Okay, another direction. So polynucleotide, next one is polynucleotide kinase. So in case of polynucleotide kinase, it will add hydroxyl group to the 5' end of double stranded DNA or single stranded DNA, and it is a ATP dependent process. Then restriction enzymes, as I have already told you, these will cut the both strand of DNA at a specific recognition site, that is known as palindromic site, or that will cut will result in the blunt or sticky ends. Okay, then reverse transcriptase, we have discussed about it. Then next one is important is tag polymerase. It is also a type of DNA polymerase enzyme, but it is a heat stable DNA polymerase that is obtained from Thermophilus aquaticus in Thermophilus aquaticus bacteria, which enzyme operates at 72 degrees Celsius and it is stable up to 90 degrees Celsius. And in case of polymerase chain reaction. Okay, so these are the few enzymes and their functions. Hope you have understand. Don't worry, you will get this table in the textbook of biotechnology by CK Dubey. You will get this. If you don't have, you can ask me, I will send you. Then we will discuss main topic, that is polymerase chain reaction. So let's discuss about the polymerase chain reaction. So if imagine there is any crime, for example any murder spot, and and police officer came there to investigate that murder. So in the past there was a one drawback for obtaining the genetic fingerprints from the material present at the crime scene. So for matching to identify the your accused, you have to take samples of genetic fingerprints or genetic material. You can take like blood spot or blood stain, then your hairs. They are used to collect that and they will analyze the DNA. Okay, but what happens, the quantity of that recovered DNA from crime scene is very less, so it was not easy to analyze that DNA and to identify the accused. So there was a new technique called polymerase chain reaction, which was developed in 1983 by Carey Mullis, which providing the breakthrough that allows the scientists to produce multiple copies of DNA sample within a very short period of time, within 1 hour. So DNA polymerase chain is nothing but the DNA polymerase chain reaction is nothing but it is a set of reaction which we using which we can produce, which can amplify, we can multiply the DNA molecules. Okay, if you have simple short fragment of DNA, you can multiply the DNA fragment into billions of copies. with the help of this polymerase chain reactions. Okay, so polymerase chain reaction is nothing but it is a similar to your DNA replication. It is similar to your DNA replication, but DNA replication occurs in the in vivo, that means inside the cell or inside the body, and but this polymerase chain reaction occurs in the lab in vitro. Okay, that is the main difference. And the enzyme used in polymerase chain reaction also has heat stability. Okay, so normal DNA polymerase will not have heat stable. It is not heat stable. It will not operate more than say 50 degrees Celsius. So DNA polymerase, so here enzyme, which enzyme is used in this polymerase chain reaction? We use here tag DNA polymerase, that is obtained from Thermophilus aquaticus. Okay, that is a heat stable enzyme. So it is nothing but the multiplication or amplification of DNA. What is the polymerase chain reaction? It is nothing but the multiplication or amplification of your DNA of interest into billions of copies within a short period of time. It is known as DNA polymerase chain reaction. This technique involves heating of DNA with high temperature so that there is separation of two strands, and then you have to cool down that reaction mixture, and with using DNA polymerase or the enzyme tag DNA polymerase, you can create a new copies like replication of DNA. Okay, so due to high temperature required for the technique, the thermostable DNA polymerase had found to be avoid the expense of needing the replenish the enzyme after each round of DNA replication. So the technique is in vitro, and it requires heat. It is involves the heat. So normal DNA polymerase will not operate this reaction. So you will have thermostable. You will have to require thermostable DNA polymerase. So tag DNA polymerase, that is Thermophilus, that is tag DNA polymerase obtained from Thermophilus aquaticus, which is a heat stable DNA. So it will lead to invention of that tag DNA polymerase. The solution of this problem was tag DNA polymerase, which is obtained from Thermus aquaticus bacterium. That the bacteria is native to hot spring, and this enzyme is able to withstand with high temperature up to 95 degrees Celsius. It means tag DNA polymerase is a heat stable. Okay, tag DNA polymerase enzyme is a heat stable, and DNA that's why it is used in polymerase chain reaction. Okay, so principle. What is the principle of polymerase chain reaction? The purpose of polymerase chain reaction is to make the huge number of copies of genes. Okay, so you have a single gene, you have to make a billion copies of that gene, then you have to use PCR. So this is the principle of PCR. It is necessary to have enough starting template for sequencing. So if you want to carry out the polymerase chain reaction, it is important to have a starting DNA template. Okay, then most of the PCR methods typically amplify the DNA fragment up to 10 kb. So most of the polymerase chain reaction uses DNA starting the DNA template of 10 kb. At least 10 kb DNA should be there for polymerase chain reaction. So PCR is used to amplify the specific region of DNA strand. Okay, so next one, what is the basic principle, basic setup, and what is the reaction mixture? So for the polymerase, to carry out the polymerase chain reaction, you will have required certain things. Okay, so we have a list of these. First of all, you will require DNA template to be amplified. Okay, जो आपको DNA amplify करना है, जिसकी copies बनाना है, वो DNA आपको लगेगा. Second thing, you will require 2 primers. ठीक है, primers are nothing but it is a short fragment of oligonucleotides. Okay, एक 20 या 30 oligonucleotide का fragment रहेगा, short DNA. Okay, so that is required as a primer. So DNA template, 2 primer, and the third one is your enzyme. So which kind of enzyme? Either DNA tag DNA polymerase, which is obtained from Thermus aquaticus bacteria, or PFU DNA polymerase, that is obtained from Pyrococcus furiosus. Okay, then fourth ingredient of your reaction mixture is your nucleotides. Okay, adenine, guanine, thymine, cytosine, your 4 nucleotides, because you will require these for the DNA is nothing but the polymer of nucleotides. So you have to you have to add the 4 nucleotides in the reaction mixture. So your reaction mixtures finally consist of DNA template, 2 primers, then tag DNA polymerase enzyme, then deoxyribonucleic triphosphatate base pairs, all 4 base pairs, then your buffer solution. Okay, this is the thermal cycler. Okay, you can see in this image, this is a thermal cycler. The instrument consists of your programmed thermal cycling. Okay, so इसमें आपको एक प्रोग्राम रहेगा जो जो आपको टेम्परेचर इंक्रीज करना है, डिक्रीज करना है, मेंटेन करना है, उसके लिए वो एक सॉफ्टवेयर मतलब प्रोग्राम्ड रहेगा वो इंस्ट्रूमेंट. ओके, और you have to take this reaction mixture in such a tubes called as polymerase chain reaction tubes. Okay, so ये ट्यूब्स होते हैं, इसमें आपको रिएक्शन मिक्सचर लेना है. ये ट्यूब्स बाद में आपको you have to mount these tubes into thermal cycler and carry out your polymerase chain reaction. Okay, so this is the reaction mixture. एक बार इस पे क्वेश्चन आया था, which one is not component of reaction mixture, so you have to take care of that. You have to remember the component of reaction mixture. Next one is cycling. The cycling process is having following step. So what are the different steps that are involved in your polymerase chain reaction? So you can easily remember these steps. There are only 3 steps. First one is a denaturation, second one is annealing, third one is your extension or synthesis. Okay, so first one is your denaturation, second one is your annealing, third one is your extension or synthesis. So what happens in denaturation? Denaturation means there is a separation of 2 strands of DNA or melting of DNA, which is usually carried out by maintaining the temperature at 94 or 95 degrees Celsius. So initially you have to increase the temperature at 94-95 degrees Celsius so that the double stranded DNA will melt and it will separate. So double stranded DNA will separate into 2 strands. So now you have separated the DNA. Now second step is annealing. Annealing means to join. So what to join? So you have to join the primer which is already present in your reaction mixture. We have already taken reaction mixture, in that there is a primer also. So okay, so you have to decrease the temperature. You have to decrease the temperature up to 54 or 55 degrees Celsius. You have to decrease the temperature from 95 degrees Celsius to 55 degrees Celsius, so that whatever primer is present in the reaction mixture, that will bind to that a single stranded DNA. Okay, now you have single stranded DNA. Initially you was having double stranded DNA, then you have separated the strands. Now you have single stranded DNA. The primer will bind to that DNA at 3' end as well as 5' end. Okay, their bonding involves ionic and hydrogen interaction, etc., etc. So you can. Third step is extension or polymerization or synthesis. So extension means you have separated the strands, you have attached the primer. Now you have to elongate that primer. You have to synthesize you have to add the base pairs into the primer. Okay, so during this step the temperature is maintained at 72 degrees Celsius. So why 72 degrees Celsius? Because it is an optimum temperature for tag DNA polymerase. So at this temperature DNA polymerase will do his work. And what is his work? It will add the base pairs. It will take the base pair from that reaction mixture and it will add to the primer, and it will add to the primer end, which will be complementary to the opposite end. Okay, so in this way the DNA will be synthesized. Your DNA will be synthesized. Don't worry, we have animation. We will. So the base pair will be coupled to the primer at 3' end. Okay, so this is the extension is nothing but the your synthesis of actual DNA complementary to the main parent DNA strand. Okay, so this is a overview of polymerase chain reaction. You have single gene, then single gene means DNA template. Okay, then you have to subject the DNA template to the first cycle of polymerase chain reaction. You will get the copies 2 raised to 2. Second cycle you will get the copies of 2 raised to 3. In the third cycle you will get 2 raised. It is a exponential amplification of DNA. Okay, in each cycle your DNA copies will be doubled. Okay, so ideally there is around 35-38 cycles for polymerase chain reaction. So you can see in this animation, first one is denaturation. You can see in this animation how there is a denaturation, how there is a annealing, how there is a extension. Means total 3 steps of polymerase chain reaction you can easily understand using this animation. So these colored dot are nothing but your nucleotides or base pairs. Okay, then this yellow colored is your enzyme, that is tag DNA polymerase. This is your DNA molecule. Okay, this is your DNA parent DNA molecule. Then these short fragments you are seeing, these are nothing but the primer. Okay, primer, then your DNA molecule, then your this enzyme tag DNA polymerase, and these are the dots of your base pairs, adenine, guanine, thymine, cytosine. Okay, so you can easily understand these 3 steps by this animation. Let's see. So first one is denaturation. Denaturation is done at 94-95 degrees Celsius to stand. Then second is annealing. Annealing means primer will attach to that your single strand. Then there is a extension. Extension means polymerase will add base pairs. Okay, so you can easily see in this animation. See, this is denaturation, means separation of 2 strand. Okay, then primer will add it to that, then your enzyme will be added and extension will be done. Okay, so this is overall your polymerase chain reaction. You can also, the technique can be also demonstrated by using, for example, this is your double stranded DNA. Okay, so this DNA you have to take in a mixture along with your DNA tag DNA polymerase, your primer and your base pairs. Then subject that mixture to 95 degrees Celsius so that there will be separation of 2 strands like this. Once 2 strands are separated, you have to decrease the temperature. Okay, after decreasing the temperature, you have the primer, whatever primer is already present in the reaction mixture. The primers are nothing but the short synthetic DNA fragments that are complementary to the DNA strand to be copied. Okay, so after decreasing the temperature, your primer will be bound to that 3' and 5' end of your parent DNA molecule. Okay, the temperature you have to decrease up to 55 degrees Celsius. Once primer is bound to your parent DNA, then your enzyme will come in action. Your enzyme tag DNA polymerase will come in action and it will add the base pair. You have to again increase the temperature up to 72 degrees Celsius so that your enzyme will be active. When you will increase the temperature up to 72 degrees Celsius, your enzyme will come in action and it will add base pair like this. It will add base pair like this. Okay, so your enzyme will be add base pair which are complementary to the parent DNA strand. So that your DNA will be new DNA will be synthesized. So first time you have only one DNA. Now you will have 2 DNA. And see in this image, 2 identical copies of target DNA sequence which result from first synthesis of cycle. Okay, so if you have one single DNA, you can get 2 DNA after first cycle. In the similar manner, now this reaction consists of 2 DNA. Now this is subjected to again for the polymerase chain reaction cycle. Then 2 DNA will be copied in the similar manner, similar steps. 2 DNA will be separated into 2 strands. Okay, there is separation. Then again you have to cool the sample up to 52 degrees Celsius so that primer will bind to that. That is a binding of primer. Then you have to again increase the temperature up to 72 degrees Celsius, so that there will be elongation or synthesis. And at the end of second cycle you will get 4 DNA. Okay, there is a exponential synthesis of DNA in the polymerase chain reaction. After 2 cycle you will get the product of 3 cycles. So there is exponential synthesis of DNA, nothing but. So it is summary. There is a separation. First of all, there is a separation of reaction mixture. You have to heat the reaction mixture up to 94 degrees Celsius. Then you have to add, and then you have to decrease the temperature up to 52 degrees Celsius, so that primer will be bind to the polymerase chain reaction, your separated DNA. Then you have to increase the temperature up to 70 degrees Celsius, so that two identical DNA will be formed at the second cycle, then third cycle, then fourth cycle, so that you will get billions of DNA after 35. And normally 35-37 cycles are done for the polymerase chain reaction, and up to the end of 35 cycle you will get around billions of DNA. Okay, so this was the polymerase chain reaction. And so what are the applications of polymerase chain reaction? So there are a few applications. For example, diagnosis of pathogens, then your identification of any mutation is there in your genes, then DNA fingerprinting is there, then in the research, then molecular archaeology, then your diagnosis of plant pathogens. So diagnosis of plant pathogen, then your molecular archaeology, then your research, then DNA fingerprinting, then parental diagnosis, then your diagnosis of pathogens are the few applications of polymerase chain reactions. Hope you have understood that this polymerase chain reaction, it is very simple. It is a very simple technique. Okay, thank you very much.